Okay, good afternoon and welcome to Let's Chat About, the free monthly webinar series hosted by Hope and Focus. Here at Hope and Focus, we've developed the series with those living with LCA and IRDs in mind, but it is open to anyone who is interested in what's happening in our communities. My name is Courtney Coates, and I'm the Director of Outreach and Development for Hope and Focus. Before we get started, I just want to take a moment to thank our sponsors of this series. They are AGTC, Dominion Energy, Editas Medicine, Janssen, Mira GTX, and Spark Therapeutics. And we could not provide programs such as these without their support. Just a few housekeeping items before we start. The session will last about 45 minutes. Your microphones have been muted and your cameras are off. You can submit questions through the Q&A function at the bottom of your screen. And as time allows, we will take any questions that come in live. We are recording this session, and after the webinar, we will post the recording to our YouTube channel and our website. It is my pleasure today to introduce Kara Fick and Dr. Shannon Boye from Atsena Therapeutics. Thank you both for joining us today. Can you take a moment and introduce yourselves and tell us a little bit about your background and roles at the company?
Yeah, sure. So thanks, Courtney, and hi, everyone. My name is Kara Fick, and I'm the Director of Patient Advocacy at Atsena Therapeutics. I live in Texas, and I've been working as a rare disease patient advocate in the biotech/pharmaceutical world for close to 10 years now. I studied biomedical sciences in college, but I am not a scientist. I leave that to Shannon and others that are much more capable of that. But I love to hear about the amazing complex science and research that's taking place. And I love to talk to families and patients that are living with rare diseases and kind of figure out how to bridge the gap so that the science and the research that's being done are meeting the actual needs of those that the research is being done for. So although it hasn't been long, it's already been such an incredible pleasure to work with Hope and Focus, and I'm looking forward to getting to know and working with this community more.
Hi, everyone. I'm Shannon Boye. I'm co-founder and director of Atsena Therapeutics. I'm also a professor in the Department of Pediatrics at the University of Florida and the associate chief of the Division of Cellular and Molecular Therapy. So I actually came to the University of Florida from upstate New York. I was obviously sick of the cold weather, so I moved to the palm trees. I came down here for graduate school, and my thesis project was actually to start developing a gene therapy for the form of LCA that is the focus of today's webinar. So it's been extremely rewarding for me to watch a project go all the way from bench to bedside. It's been really exciting. I don't think many graduate students can say that. But now I'm dating myself, but I've been at the University of Florida for over 20 years, focused on developing gene therapies for inherited retinal disease. And I have to say, I've got to give a shout out to the Foundation Fighting Blindness. They were the very first foundation or entity period to kind of take a gamble on me and to fund my research. And with their support, I was able to generate the preliminary data necessary to compete for a larger grant from the National Institutes of Health. So I really attribute the FFB to really getting my independent research career started, and all of that kind of spiraled in ultimately to the formation of Atsena. So I'm really happy to be here to talk with you all and tell you our story.
Thanks so much, Karen, Shannon. It's really great to have you both here today. And Shannon, your extensive history with LCA1 is pretty exciting. Can you tell us a little bit more about how Atsena was founded?
Sure. So as I mentioned, I've been at UF for about 20 years. I met my husband in graduate school, so it was a total nerd romance, as you can imagine, in the lab. And I actually met him because I needed help cloning one of our Gucci 2D plasmids for reasons I won't get into right now, but it's a difficult thing to clone. So that's how we met. I still have the little Eppendorf tube in my jewelry box. But we worked together for many years, and we have a history kind of generating these technologies that we thought at the time, okay, the best path forward to get these technologies to patients is to partner with an entity that already exists. So to give our technology to a company that already was built, that had the infrastructure and the capital to move it forward. And so we did that over a number of years, both with large pharma, small companies, medium companies, you name it. And that was really rewarding on some levels because we got to network with kind of everyone doing ocular gene therapy on the industry side. But one common theme that emerged for us was that we were frustrated because the therapies that we were developing were not making their way to patients in as timely a fashion as we had hoped, if at all. And there were multiple reasons for that. In big pharma, as many of our listeners might imagine, you have different functions and these huge companies all siloed apart from each other, and they don't talk to each other very effectively. So, they were just going very slowly was the bottom line, and that was frustrating. And then in some of the smaller companies, unfortunately, we saw business decisions overriding sound scientific decisions. So it got to the point where my husband Sanford and I were just frustrated. And so one day I sent, I call it a Shannon email because it was kind of a big long vent to the then CEO of the Foundation Fighting Blindness, Ben Yerxa. And I said, why is this entity doing it this way? Why is this entity doing it this way? I'm so frustrated. We should be doing it this way. And he wrote me back, let's chat. It was a very short response to my long-winded email. So we got on the phone, and he was the first person really to push me over the edge of starting my own company. I had thought about it, but I was a little nervous about it. And he really mentored me through the process of what was required to get a company off the ground, and walking me through, the technology that we had and the IP that we had. And he was just incredibly instrumental in kind of helping Atsena to form. So the Foundation Fighting Blindness's RD fund was instrumental in getting Atsena off the ground, as was a number of other smaller investors in the earlier stages. So that is kind of how Atsena formed. It formed out of frustration, more or less.
However you came to be, we appreciate you being here, and we appreciate the work that you're putting into this trial. It's pretty exciting to see that come that whole 20 years for you.
Thank you.
I'm going to turn it over to Kara now. I would love to hear a little bit more about how your role plays into Atsena.
Yeah, sure. So I think it's pretty apparent that, from Atsena's founding, really keeping patients at the center and trying to move research forward so that it can get to patients is super important to Atsena. That starts with Shannon and trickles all the way down through us. So in my role as a patient advocate at Atsena, I'll be listening and learning from the community about what's most important to you, and trying to bring some of that information and those insights back into our company and make sure that the research that we're doing is reflecting what's meaningful. So I'm really interested to learn and hear more about things like, your journeys to getting diagnosed, things that you would or wouldn't want out of research or out of clinical trials, and just really the day-to-day aspects of living with LCA. All those things that you guys are living through day to day, but aren't ever captured into a textbook. So I'll be sharing all that back with our company, but I'll also be coming to you guys and sharing information about what Atsena is doing, and really just trying to make sure that this is a two-way flow of information and learning back and forth between the LCA community and Atsena as a company. Now, I will say, like I said, the passion for making sure that we are serving patients starts at the top. I joined Atsena about 4 months ago, and the company is still a pretty small biotech compared to many others. When I joined, there were only about 35 employees. We've added a few more since then. And from what I've seen in my career, that's really, really early on to hire a patient advocate. And I think it's pretty unique that Atsena has prioritized bringing someone in as a full-time patient advocate at the company so early on, because we all talk about how important it is to fold in the patient perspective, but to actually show that and take action and bring me in so early on, I think just really shows that we're a pretty unique company. And many are in this space. There's something very special about the inherited retinal disease space, and many others that we've met here, but it's just, it's fantastic to be able to come in super early on, make sure that that patient perspective is incorporated into everything as early as possible.
Thanks. I know your role has been, incredibly important for us so that we have a lifeline with you guys. It's just really nice to have that dedicated person who is looking out for our community. So I really do appreciate that. Heading back to Shannon, you spoke a lot about your history of working with LCA since graduate school. Has it always been with LCA1, or have you looked at other forms of LCA or IRDs?
Yeah, I've focused on a number of different indications. Specific to LCA, I've looked at the CEP290 form a little bit more peripherally. My main LCA focus, though, has been on the Gucci 2D form, so LCA1. But outside of LCAs, we've focused on XLRS, on Usher syndrome 1B, on autosomal dominant cone rod dystrophy, a number of different indications. And then in addition to the specific indications, what we work on a lot in my lab and now also in Atsena is optimizing the tools with which we do the gene therapy. So constantly making bigger, better, faster, stronger gene therapies that we can deliver to these patients.
Awesome. So what would a gene therapy look like for LCA and how would that work? Can you touch on that a little bit for us?
Yeah, definitely. So to explain gene therapy, I like to take people generally back to around high school level science, but not like senior year high school level science, more like freshman year. So we're okay, don't worry. But all of us have genes, and your genes are different from my genes. That might be why you have blue eyes, I have brown eyes. That's a simple example, but all of us have genes, and our genes do something important, and that's that they make proteins. And you can think of proteins like the building blocks of life. So proteins are acting alone or in combination with other proteins at every second of the day and night in order to perform essential functions in your cells. So a good example of proteins that are really important for vision are the proteins in your photoreceptors in your retina. And those proteins all work together in order to convert light into an electrochemical signal that's sent to your brain and processed there as vision. So genes make proteins and proteins perform essential functions. But sometimes we can have a misspelling in our genes, in other words, a mutation. And as a result of that misspelling or mutation, the protein that gene was supposed to make doesn't get formed. Or in other instances, maybe the protein gets formed, but it's misshapen and it can't interact properly with the other proteins it's supposed to talk to. So when that happens, you don't have a protein present in, for instance, your photoreceptor cells. And then all of a sudden your photoreceptors are no longer capable of transmitting light into a signal that's processed as vision. So genes make proteins, proteins perform essential functions, mutations can lead to the absence of that function. And so the concept of gene therapy is actually pretty simple. It involves taking a healthy copy of a gene that lacks that misspelling, so it's got a normal spelling, and delivering that gene to the photoreceptor cells. Once inside the photoreceptors, that gene can go on to make the protein it was supposed to make and hopefully restore function to those cells. So genes make proteins, proteins perform functions. If there's a misspelling in the gene, you deliver the healthy gene, it makes the protein, it performs the function. So that's gene therapy as a whole, but you might be wondering kind of how do we get that normal gene into those photoreceptors? So we use something that might sound scary initially, but we actually use a virus. And it sounds scary because viruses usually make us sick, but when you think about what viruses do for a living, it makes perfect sense. We're actually harnessing nature in a sense. So when a virus enters our body, it infects our cells and then it releases its genetic information. And that's exactly what we need it to do in order to perform gene therapy. But first, we gut that virus of all of its native genes. So there's no way we're going to be delivering anything harmful about that virus to a patient. We gut it of its native genes and we replace it with a healthy copy of a gene like Gucci 2D, for instance. We then deliver that vector, or that virus, to the photoreceptor cells. It infects the photoreceptor cells and drops off that healthy Gucci 2 gene, which then goes on to make the protein it's supposed to make. In this case, it's called guanylate cyclase 1. And then that protein restores function to the cell. A really important component about gene therapy is that in order to have a positive impact, the photoreceptors still need to be present. So if there's not a cell to drop off that gene to, then we will not have therapy. In LCA1, though, we might get into this in a little bit, but we have the fortunate case where these patients do maintain retinal structure and photoreceptors over their lifetime. So these patients are extremely good candidates for gene therapy. From there, do you want me to just go over kind of the preclinical work that we've done on this indication, Courtney? I don't want to.
I'd love to hear more, knowing how traditional AAV gene therapy works is, really important. And, seeing how did that inform your clinical trial and prepare you to take LCA1 to the next step?
Sure. So having a virus that can deliver a gene is one step of the process, but another really important point is that you need a good animal model of the disease. So what that means is an animal model that mimics the patient's condition. So in LCA1, you have patients that have profoundly impaired retinal function. So their cone photoreceptors, which are responsible for visual acuity and color vision, are profoundly dysfunctional from birth. And they have variable levels of rod photoreceptor function. And we use our rods under dim light conditions. And Kara will talk a little bit more about this later. But these patients are typically diagnosed at birth or within the first few months because parents notice that their babies aren't fixating, or maybe they have a roving eye movement. But it's associated with profound retinal dysfunction. But as I mentioned, very good preservation of retinal structure. So we needed to find an animal model that mimicked that patient phenotype, that LCA1 phenotype. And we were fortunate actually that there are multiple models of this disease. I actually started working on a chicken model of LCA1, which was entertaining and I won't get into, but we moved very quickly into mouse models of the disease, which are much more translationally relevant because they are mammalian, just like patients. So much like the patients, they had retinal dysfunction, but pretty good preservation of retinal structure. They also had very impaired visually guided behavior. You can actually do behavior tests on mice to determine that. And so the next step for us was finding the right flavor of AAV, which is the virus that we use in order to efficiently deliver a healthy copy of Gucci 2D to those mouse photoreceptors. So we spent a number of years kind of looking at a variety of different flavors and regulatory units that were packaged into that virus and found the ideal combination. And that was tested all the way up into non-human primates, which are a clinically relevant species in the sense that they have that very specialized central cone-rich retina like patients do. So once we had the unique AAV combination, it was time to treat the animal. And so we do what's called a subretinal injection, which is a similar procedure to what's being done in the clinic in patients. And then we wait a few weeks and we perform a test called an electroretinogram or an ERG. That's the same type of test that's done on patients in the clinic. And we found that when we treated the right eye, for instance, with our gene therapy, the right eye had very robust retinal response in the ERG test, whereas the untreated eye was flatlined. So we immediately saw very significant improvements in retinal function. We saw preservation of retinal structure. We saw restoration of visually guided behavior up to normal levels. So these treated mice were behaving just like wild type sighted mice. So at that point, we partnered with a company prior to Atsena, and we did all of kind of the big necessary preclinical studies to bring it from bench to bedside. So not just kind of lab experiments, but like the really hardcore documented IND enabling studies that are necessary to bring a drug all the way into patients. So that was done under the umbrella of a company called Genzyme, which was bought shortly thereafter by Sanofi. And unfortunately, Sanofi decided at a certain point that they were going to move away from ocular gene therapy. So they had another couple of programs that weren't working very well, but that was good for us because we were able to bring the program back. It was kind of a homecoming when it came back to Atsena, to the founders that had developed the gene therapy initially. So I think that answered your question. I'm sorry if that was a mouthful.
No, and just to put everything in perspective a little, can you just comment quickly on what's the time frame we're looking at for your preclinical work? How many years did that all take?
So I would say we started working in earnest on the mouse models in 2004. We partnered with Genzyme Sanofi in about 2012, and that's when we had kind of a period where things slowed down a bit. I have to say, though, that there were benefits to that because we built such a strong preclinical package in that partnership with Sanofi that by the time we got to clinic, there was no question about what was the minimum effective dose and what was the highest tolerated dose. So that partnership was amazing. Did it go a little bit more slowly than I would have hoped? Yes, but you know, the rest is history and we are where we are and things are working now. So I wouldn't change it. But it was a long road.
Yeah, it does take time to solidify the science, but, you are in a clinical trial now. So maybe you can speak a little bit to what phase you're in and, what does that look like?
I will hand that over to Kara.
Sounds good. I'll take it. So drug development and gene therapy development, it takes a long time, and there's a lot of processes that we have to go through to make sure that anything that is given to patients ultimately is going to be safe, and it's also going to work. So after all of the discovery work and the preclinical studies in chickens and mice and monkeys, or whatever other animal studies are done, then there are usually 3 different phases of clinical trials in humans that a potential medicine, or in our case, gene therapy, has to go through before it can be considered for FDA approval, or approval by the equivalent regulatory bodies in other countries. So there's 3 phases, but sometimes for rare diseases like LCA, those 3 phases can be combined a little bit. The earlier phases usually focus primarily on safety and figuring out the best dose. So the minimal dose that can be given while still being the most effective. And then the later phases focus a little bit more so on testing the efficacy, or how well it's actually working. So Atsena right now is working on a combination phase 1/2 clinical trial for LCA1 caused by mutations in the Gucci2D gene. And as a phase 1/2, this trial is really meant to test the different doses of our gene therapy, find that best dose, and to closely monitor the safety of the gene therapy. Now, because we're doing a combo phase 1/2, we're looking primarily at safety, but we're also incorporating some tests into the clinical trial that can help to tell us a little bit about the efficacy of the gene therapy as well, give us kind of a hint into that portion. So fortunately, as a rare disease, there are benefits that you can do to kind of speed up this whole drug development process, and being able to do one of these combination phase 1/2 clinical trials is one of those efforts to speed things up, because we know that every day is important. And, had this research been done yesterday, it still wouldn't be soon enough. So we're trying to do everything that we can to make things faster and still make sure that everything is safe and effective.
Yeah, and just for clarification, what are the age ranges that you're looking at this phase of the trial?
Sure. So with this phase 1/2 trial, again, it's focused mainly on safety. So we started it off with adults age 18 and up. But towards the end of the trial, with the last few people that we were open to enrolling, we did open up that age range down to pediatrics, so down to age 6. So overall, if you're looking at the full trial, we enrolled and we were open to enrolling anybody age 6 and up, depending on what part of the trial they were enrolling in.
Great. And, how's your progress there? Is there anything you can share?
Yeah, sure. So this phase 1/2 trial, it was designed to include a total of 15 participants all with a Gucci2D mutation, and test different doses of our gene therapy. So we were testing a low dose, a middle dose, and a high dose. And each patient that participated would receive the gene therapy in one eye through subretinal injection, like Shannon mentioned earlier. What we're looking at with this trial primarily is the data that would be seen 52 weeks or 1 year after receiving the gene therapy treatment. So we're looking at all the different results that are seen primarily within that first year. But we're also going to be following people for a total of 5 years to make sure that we're paying attention to long-term safety and doing long-term follow-up and monitoring throughout those 5 years. So at this point with our trial, we have enrolled all 15 participants, so the trial is fully enrolled. But the various tests and data collection, that is still ongoing for the trial. So it's not wrapped up, it's not finished, but we're moving forward and happy to have all 15 people who have chosen to participate. There's one thing that I'll mention with this phase 1/2 trial, and likely for our future trials as well. We have what's called a data monitoring committee. And this is a group of experts that review all of the safety data throughout the trial and look at it really closely to make sure that there aren't any type of safety concerns that would lead us to stop or not move forward with giving the gene therapy to the next person in line in the clinical trial. So this data monitoring committee would meet every time we decided to up the dose. So they met before moving from the low dose to the middle dose, and they met again before moving from the middle dose to the high dose. And they continue to meet on a regular basis, always constantly looking at the safety data so that we're paying super, super close attention to that safety of the treatment.
Great. And any preliminary findings so far that we could hear about?
Well, what great timing that you asked this question, Courtney. Atsena actually hosted a presentation just at the beginning of this month, so it's kind of hot off the press right now with some of the initial data from our phase 1/2 trial. So we presented this at a big meeting. It's called the American Academy of Ophthalmology Scientific Conference. And I'll do my best to try to pack it all into a nutshell and summarize it as best as possible. So you asked earlier what the age range open to the trial was. The actual ages enrolled in the trial, so we had 15 people that were between the ages of 12 and 76. So we had a pretty broad range of ages that participated. Again, we did allow participation as young as 6 years old, but the youngest actually enrolled was 12 years of age. And I'll just kind of put a reminder again that before I get too far into the results, not everyone has gotten through an entire year after being treated. So this is just preliminary data, and we still have more that we have to collect, analyze, and summarize before we have the full results of the clinical trial. So as a phase 1/2, I've said it many times, this trial's first priority is safety, so I'll talk about that first. The gene therapy is administered as a subretinal injection, and with any surgery, there's always some risks associated with that, but we're really pleased that we didn't have any reports of serious side effects. And when I say serious, that's defined as requiring hospitalization. So we did not have any reports of serious side effects that were determined to be related to the gene therapy. So that's encouraging to us. And nobody has decided to drop out of the clinical trial because of side effects. What we have seen are 2 cases of inflammation, but those seem to be pretty minimal and were resolved after being treated with steroids. So all in all, thus far, it seems that the gene therapy is safe and well tolerated, but of course, we'll continue to monitor safety and collect more data throughout the trial to ensure that that continues on. But so far, things are looking to be safe from what we've seen. I mentioned earlier that it's a combo phase 1/2, and we're looking a little bit at efficacy also to give us a hint. So we're doing all sorts of different tests beyond just safety monitoring to kind of see what sort of effects we're seeing from the gene therapy. Lots of different tests, but I'll just highlight 3 of them. And those were presented at this conference earlier this month. So the first one is a test called FST. That stands for Full Field Stimulus Test. And with this test, essentially, a patient will put their head into this dome machine, and the dome will flash different colors and different brightness levels, and the patient will push a button to indicate whether or not they saw the flash. And with this test, we did see significant improvements for those individuals that received the highest dose of the gene therapy. So we're really encouraged seeing those improvements early on. The second test that I'll talk about is called the MLMT. And some of you may have heard of this because Luxturna really made the MLMT kind of a famous measurement with clinical trials. And if you haven't heard of MLMT, essentially it's a small obstacle course, small maze with obstacles that a patient is asked to navigate through at different light levels. So bright levels, dark levels, and kind of see how they perform navigating through this maze. This was a test that we didn't actually have in place at the beginning of our clinical trial. We kind of brought it in a little bit later. But the people that have been able to complete the test did show a clear improvement before and after receiving the gene therapy treatment. Or if you compared participants who went through the MLMT using just their eye that was treated with the gene therapy compared to the eye that was not treated, we were able to see improvements. The third test that I'll mention is probably the more stereotypical test that people think of with any type of trial that's related to vision or just day-to-day measurements of vision. It's called BCVA, and that's the poster that has different letters that you're asked to read out. And what that's doing is measuring visual acuity, so how well of detail you can see. Now, when we looked at this test, there were some patients who had some improvements, and there were some patients who didn't. So the response seems to be kind of variable on that. But we know that this is a test that the FDA finds really important. So we're going to continue to collect data and keep on administering this test and see what sort of results we see from BCVA. So those are the three primary tests I'll talk about with the clinical trial that we've got some results for so far. There's lots of other things being done, lots of exploration being done at this stage. But I'll also mention that we're collecting some patient-reported outcomes during the trial as well. So there are different surveys that have been validated to ask patients directly what they are experiencing with their vision. And we're collecting that to hear directly from patients how their vision may or may not be changing before and after receiving treatment with our gene therapy. Lots packed into what I said was going to be a nutshell. It's a very large nutshell.
No, we really appreciate the detail, Kara. I know that it's very exciting to hear this preliminary data being positive, and that it will hopefully set a good foundation for you guys moving forward. And as exciting as it all is, we have to ask, what's next for what I'm assuming is phase 3.
But of course, so it's definitely very encouraging what we're seeing. The fact that there haven't been any major safety issues, and we're seeing some of these initial signs of efficacy. But again, this is still just the preliminary data. Next steps, we need all 15 participants in our trial to get through their first full year after being treated with the gene therapy, since that's the primary thing that we're looking at is results after 1 year after treatment. So once all 15 participants get through that full year, then we'll need to collect all of the data, analyze it, and really summarize it all very vigorously and thoroughly in order to consider moving on to the next phase. So we're starting to do some things that will help us plan for the next phase, but we have to get through all of this data. We still have more data to collect before we can move forward. We're also still going to need to meet with the FDA and with other regulatory agencies in order to get their agreement and permission, essentially, to move forward the way that we're planning to. We want to make sure that if we move into a phase 3 trial, it would be designed in the way that the FDA would agree with and would find meaningful, and other regulatory agencies as well. I say the FDA because we're here in the US, but we're certainly not only purely focusing on the US. So finish our current trial, talk to the FDA and other regulatory agencies, and then we'll be able to move forward into launching a phase 3 trial. Sometimes that's called a pivotal trial, but it's a phase 3 trial. And so, as I mentioned earlier, phase 3 is really more so focused on efficacy and seeing how well the gene therapy is working. It'll still pay attention to safety, but the primary goal of a phase 3 is to focus on efficacy and use that as supportive data that could be submitted to the FDA for approval, to actually get full approval of the gene therapy for widespread use outside of a clinical trial. I will make sure that as we have more updates, I'll be working very closely with Courtney and with Hope and Focus, and we'll share any information, any updates that we have with the community, and make sure you guys are all kept up to speed with anything that we can share as soon as we can share it.
Yeah, and we appreciate that. And vice versa, is there anything that Hope and Focus and the LCA community can do while you're working on this and trying to forward this research?
Absolutely. So I think, the first thing that people think of, the obvious thing is, oh, I have to participate in a clinical trial in order to contribute to research. And that's great, that's important, but there's so much more that the community can be doing outside of that if that's not the right option for you, or it's just not the right time, or it's not available to you. So the first thing I would say is getting genetic testing is becoming more and more important, and figuring out the specific genetic mutation that may be causing your LCA. As more trials are starting, more research is being done that's gene specific, it's going to become more and more important to know exactly which genetic mutation may be causing your LCA. So I would say, absolutely, if you have not been genetic tested, talk to Hope and Focus, talk to the Foundation Fighting Blindness has a genetic test that they support so that there's not a cost to patients and family members to receive genetic testing, and hopefully remove that as a barrier. The second thing I'd say is an empowered, active community goes such a long way in research. So even just tuning into this webinar right now is showing that you've got an engaged community, participating in surveys that Hope and Focus may be sending out. I know they sent one out earlier this year that was really, really interesting. Getting involved in that and participating in surveys goes a long way. Going to any type of events or family conferences or meetings or anywhere that you can get involved and just show up, whether that's virtual or in person, really, really goes a long way towards moving the needle forward for research. And then the last thing I'll say is telling your doctors about Hope and Focus or patient communities like this. So if they come across someone else who has LCA, they can get them plugged in and grow this community. And, you know, there's so much that is beneficial from you all connecting with each other and being connected to Hope and Focus that contributes towards research that may not seem as direct as participating in a clinical trial, but is absolutely just as critical.
Thanks. And thank you for that plug for our survey too. I'm going to drop a link in the chat for anyone who hasn't participated in the LCA community survey yet. We are looking for those insights, and it does help us inform our partners about where our community is at. So having said everything that we have, you know, if someone were interested and wanted to connect with you both, what's the best way?
Sure. We love getting connected with the community. A few of us were able to actually go to dinner in the dark this past weekend and get to meet some of the community in person, which was fantastic, and we'll continue to be showing up to Hope and Focus events. But if you have questions, or if you want to just share your story or get connected with us, you can email Atsena at patients@atsenatx.com, and we can drop that in the chat too. But patients, as in PATIENTS, at atsenatx.com. You'll be able to get in touch directly with us. And then, you know, if you have questions for whether it's Shannon or myself, we'll make sure that it gets directed to the right person.
Great. You know, we really appreciate you both sharing all the work that Atsena is doing. It's pretty amazing, and it's pretty inspiring for everyone in our community. So really appreciate that today. I do want to try to take a couple questions from the audience since we have a few minutes. So those of you that are attending, please take a minute, drop your questions in the chat. I know we did receive one while we were speaking. Mother of a 15-year-old child diagnosed with LCA1 and has a question about when you start trial with children under 6 years of age, and just the hope that that provides some parents.
Sure. So our current trial was open to ages 6 and above, but moving forward, in order to consider allowing participants who are younger than age 6, we really need to get the full support and agreement from both the scientific side as well as the FDA side. So unfortunately, I don't have a specific answer that I can provide as to when we would be able to start opening up to younger participants or younger children. But we know that that is very important to the community. It's at the forefront of our minds, and we'll certainly be doing what we can to vet the scientific side as well as the regulatory side of that. Shannon, feel free to chime in if you've got anything else to add on.
Sure, just from a scientific perspective, you may remember me mentioning that LCA1 is fairly unique from the standpoint that the retinal structure in LCA1 patients is maintained. It's not a very aggressive degenerative condition. So that intensity to get treated immediately may not be as relevant for LCA1 as it is for other inherited retinal diseases. And we're seeing treatment effects in patients of all ages that we've looked at so far. So hopefully that lends confidence to the little bit of a wait. I know that it's hard as a parent, I'm sure, but find some comfort in the fact that retina is not going anywhere is I guess my point.
Yeah, I think that's a really positive way of looking at it. And, thankfully there is some good news there. I'll just ask a question as well. As you go through this work with LCA1, and I know, Shannon, you've had some background with other forms. Are you hoping that some of the work that you're going through will potentially help other genetic mutations?
Oh yeah, I know it will. So figuring out what flavor of AAV or what serotype it's more scientifically called, and what regulatory elements are being used to drive expression of the gene, which are called promoters, those being identified for LCA1 is going to be useful for any other photoreceptor targeted disease. And, LCAs fall almost exclusively into that bucket with a few exceptions. So for any photoreceptor indication, having identified that capsid and that promoter to drive gene expression is going to be relevant. And then, of course, all the work that Kara described on the outcome measures that we're employing, FST, for example, is an outcome measure that is receiving a lot of attention. We're showing significant gains in the retinal sensitivity with this outcome measure. And while it may not be an approvable endpoint right now in the United States, it is an approvable endpoint in other parts of the world. So, all of these tests that we do in this clinical trial are getting these different agencies more and more comfortable with these outcome measures. And we think that will be beneficial across all inherited retinal diseases.
Yeah, there's a lot of discussion across our industry with endpoints. And I think, the more we go through these trials and identify new ways of looking at endpoints, it's really going to be beneficial across all the different forms. I do have one more question that came in asking if you have any natural history studies for LCA1 and if you can discuss how important or not that is for the FDA approval.
Kara, you want me to take?
Yeah, Shannon, if you've got this one.
Yeah, sure, absolutely.
Yeah.
So to my knowledge, there have not been any, the FFB is great at sponsoring these formal natural history studies. And while we haven't had perhaps as robust a natural history study done in LCA1, we have had investigators who have dedicated their lives, like Dr. Jacobson at the University of Pennsylvania, to characterizing this disease. And so he has published multiple manuscripts looking both at snapshots of the disease and multiple patients, as well as following patients over time. So it may not be as robust as it is in some indications, but there's certainly a ton of information out there being done by multiple groups.
Great. And how does that play into the FDA course for you?
It bodes well. I mean, the more we know about the disease, the more we know what benchmarks we need to meet, right? We know what the structure looks like. So we're not looking to improve structure necessarily, but we know we're dealing with profound dysfunction, and, identifying outcome measures that can look at improvements in that function is really what we need. And that's well laid out in the literature.
Great. It's all super encouraging. Is there anything else that either of you would like to share before we close out our webinar today?
If anyone's in North Carolina, come see us.
Yes, we didn't mention that. Although Shannon and I are spread out, I'm in Texas and she's in Florida. Our office is in North Carolina, so we're trying to reach many different corners of the map as a small team. But we would love to have some visitors sometime up at our office in North Carolina.
Great. And I'll just remind everyone that if you want to get in touch with Atsena, the email has been dropped in the chat, and you can always reach out to me and I can get you connected. But we're really thankful for your time today, Shannon and Kara, for all this great information, and grateful for you doing this important work in our community. For those of our attendees who are on, if you haven't signed up for our newsletter, you can do so on our website at hopeandfocus.org. And that way you can stay in touch with everything that will be to come. I think we're going to have a lot more news coming out of Atsena.
Thank you all for your time so much.
Yes, thank you for having us, and we are loving this beautiful relationship we're starting to build with Open Focus. You guys are a great group, and excited to share more in the future.
Great. Thank you all. Have a wonderful day.
All right, take care. Bye-bye.
Bye.